All right, good morning. So my name is Ed Conway. I'm a little loud, thanks. I'm still the director of the Center for Blood Research. And I have the honor of once again welcoming you to another Earl Davie Symposium. This is the 11th one. Just a couple of logistical details, just to remind you, the washrooms are that way. All right, so beyond the registration desk, there'll be 2 coffee breaks, lunch, and at the end there'll be drinks and snacks and good stuff. All right, posters are over here to my left, just outside, so they'll be open during the different breaks, the coffee breaks and the lunch break. There'll be judging of those posters by volunteer judges. And the judges should have those scores put into Mira and Julie by the end of the second coffee break, please. There's a touch for speakers, there's a touch pad. Best to use that, okay, rather than with a big arrow. So for me, it's huge. All right, so that you can see it, and so we don't just video and webcast your right ear or left ear. And what else do I need? There are evaluation forms that everybody should have received. Please complete these by the end of the day so we can improve the symposium next time. So before I do get underway, I first want to thank the sponsors. That's the most important part. We have lots of representation from our sponsors. It's Novo Nordisk, Pfizer, Shire, Bioverativ, the Canadian Blood Services, CSL Behring, the Bayer and the Bayer UBC Bleeding Disorders Collaboratory, Octapharma, Grifols, the Naaman Vickers Hematology Endowment Fund, Alexion, New England Biolabs, and Stego. So all of these sponsors really make this event possible. Without their support, we couldn't do this. So we want to thank them. When you see them, please thank them. All right? Before I start, I just want to introduce, just so you're familiar, because some of our visitors particularly aren't familiar with the Center for Blood Research. The Center for Blood Research was founded by Ross McGilvray, who is walking in as we speak. Always on time, right? It was started in 2002, and with input from or support from his colleagues shown here: Dana Devine, Don Brooks, Grant Mock, and Chip Haynes, who really had a vision of creating a multidisciplinary research institute devoted to blood research, and it's expanded and developed into a program that cuts across all disciplines, so engineering, science, arts. Really everything, so that we have 40 investigators spread across UBC, sort of with a headquarters on campus at the medical building, and we cover all disciplines, but with a focus particularly on transfusion science, inflammation, infection, and hemostasis and thrombosis, which is really the reason we're here. But you'll see that we are a very hybrid group. Really, we emphasize sort of bottom-up training of and education and training of our trainees, that whether they be clinical or fundamental scientists, and encouraging interactions between those groups. And I think we do a reasonably good job, but we're always looking for opportunities to enhance those interactions. We're fortunate to have a very lean and mean team. Please, Ed Prysdale, Ed, wave, and Ed's the Associate Director, and he's been here from the start, from before when I was here. He's an integral part and supporter of this program particularly, but the entire center. And then we have Julie up here and Mira down here, who are our education program manager and office manager, and they are really responsible for all of the logistics. The speakers are well aware that they've really created all the opportunities to make this happen. So please, you can thank them when you get a chance. Also, all the students, we have a quarterly magazine, and you'll probably see that scattered around. Please enjoy, take it with you, distribute it, whatever. There's all kinds of stories about science, about ethics, about Politics, who knows? But the students do all the writing. I get a final edit to make sure it's not too inflammatory. But otherwise, it's their creation. They do everything. I really don't do anything at all otherwise. And it's a really a mark of their successes. So why are we here? Now, some of you have heard this story. Many of you know the story, but I like to retell it in any case. So Earl Davie. Earl, do you want to wave? Okay, here he is. Earl has had, as you probably all know, a huge, huge impact on medicine, science and medicine, that will stand the test of time, truly. I mean, there's not a patient who goes into hospital who doesn't benefit from his contributions in terms of the coagulation cascade. Every PT and PTT that we do in a pre-surgical or otherwise, is the foundation is built on our understanding of the coagulation system as he really developed it. So Earl was born in the state of Washington, and there he is as a child, building the blocks of the coagulation system from the very start. After a few years of doing that, I kept falling down. He put him up, but he persisted and then ultimately ended up doing his PhD with Hans Neurath at the University of Washington, and really he encouraged him to look at how chymotrypsin gets converted to trypsin through proteolysis, and that really set the stage for further study. So he did a postdoc in Boston and then came back to Cleveland at Case Western, which led him to interact as a young staff person with Oscar Ratnoff. And really, that's really the famous waterfall cascade came out of that interaction where he identified Hagman as a patient who had factor 12 deficiency and characterized factor 12 and started to understand the initiation of the coagulation system. And that led to a whole series of purifications of proteins in the coagulation system, which is not so different from what we understand now. But just to go back, here are a few of the coagulation proteins that he characterized from a protein basis. And then Ross McGilvery and Dominique, where's Dominique? Dominique, wave, where? There's Dominique. Dominique and Ross really brought DNA technology into Ross's lab, and into Earl's lab, and set the stage for cloning of all these proteins. So you can see no small amount of contributions just in that cascade and understanding the system, which led to a better understanding of all the deficiencies. In 1981, he spun off a company with our own Nobel laureate here, and Mike Smith. Mike Smith, thank you. It's early in the morning still. And spun up a company, and that really set up a system to recombinantly make proteins, and in particularly insulin. And then they were bought out by Novo Nordisk, and that's really the foundation of their business, which was insulin and factor 7A, actually. Another amazing contribution in terms of impacting on patients who have diabetes, which is obviously increasing the need in terms of insulin. Now, a couple of weeks ago was Earl's birthday. It was his 90th birthday. So, and Eddie was there, and I had the pleasure and the opportunity to sort of join this little party of his colleagues and many of his students who I didn't really know. Ross would have known. Unfortunately, Ross couldn't go, but Ross would have known a lot of these. And there were three things that came up during that little party. Was, first of all, the amazing support, wonderful support that he has, and they were testimonials really of his, not only of his science, which goes without saying, but how supportive he was and what a fun lab it was throughout all those years. I mean, there wasn't anything bad said. I can't understand that. All those stories. I'm sure that wouldn't be the case for me. But of course, only those who loved him were there. But it was an amazing afternoon. But the second was, he told this story about why there was so much money in cardiovascular research, when he was working in the field. And it turns out that he tells the story that this man over here, for those who aren't familiar, is Dwight Eisenhower. That's the infamous Richard Nixon. And Eisenhower was the president from 52 to 60, and he was loved. And he had a heart attack on a golf course on the 8th hole in 1955, and because the country was devastated, from what I understand, the stock market dropped lower than it had since the Depression, but at the same time, Congress introduced research funds in the scale of millions and millions of dollars towards cardiovascular research. So it had a huge impact, the health of a president. So just imagine what would happen today if the same thing happened on the golf course. That's the only political comment I'll make. Now, the other thing is that I found out was that Earl really didn't want to be a scientist, or maybe wanted to be a scientist, but he wanted to be a pilot. So we have made him a pilot here. And there he is. And time isn't too short, Earl, because I think that there's still a future for you to do more beyond just flying an airplane. All right, so now we're going to move on to the program. So as you can see, this symposium is held in Earl's honor, and really I send out an invitation to attend this conference or to speak at this conference, and really I only have to wait a few minutes before I get a response, yes, I will be here. This was Ross's idea 11 years ago to introduce this program, and it's really taken off, and we're really pleased and honored to have him to come and bring his friends and colleagues to this meeting. They certainly wouldn't be here if I invited them here. I'm Conway, please come. It's really in honor of Earl, and that's why we're here. So we're going to start the program, and the first few talks are on hemophilia. And I'll introduce the first speaker in a second, but just to give you a little background, hemophilia has undergone, as you all know, spectacular changes in the last several decades. If we look back to the 1950s and 60s, and even when I was in the clinics more like the 70s and 80s, but even at that time, the standard of care was far it wasn't exactly ideal. And in the 50s and even the early 60s, the lifespan of these kids was not measured in many decades. It was in the 20s if they were lucky, and they were they were inevitably crippled if they had any severe hemophilia. So here you can see a couple of examples of what they look like. But today it's quite a different picture. Certainly in North America and the Western world, you can see what the kids can do. They can really do anything with good care and continuous care. So we're really thankful for the scientists and the clinicians who have propelled us forward in this manner. But that's not the case everywhere, okay? So this is still 2017 in Africa, where there is a huge majority, actually, of patients who are mostly unidentified, and if they are identified, they don't have access. And I do some work in Uganda, and these are kids in Uganda. This boy who's 11 years old certainly is chronically crippled, and he's got two brothers who also have hemophilia, and they live in the city but can't get access because of transportation or whatever. So there's a long way to go, and we certainly need research in the field and advances that provide long-term therapies that are effective. And that leads me to introduce our first speaker. So we're delighted to have, let me just open up her slide. Dr. Catherine High, who is the president and head of, the head of R&D and the co-founder of Spark Therapeutics. She is focused on developing approaches to solve genetically genetic diseases using gene therapy. She's got a long, amazing history in this field. She's really a leader in the field. She was a professor of medicine at the University of Pennsylvania. She was a Howard Hughes investigator. She was a founding director of a Center for Cellular and Molecular Therapeutics, and she's, for her contributions in the field, particularly in hemophilia B initially, but now expanding further. She's been elected to the National Academy of Sciences and the American Academy of Arts and Sciences, and she's, we're really thrilled to have her here to speak to us as a first keynote speaker. Kathy.
Okay, well, thank you, and I really am delighted to be here. And of course, everything that we've done in gene therapy for hemophilia has been built on the work that Dr. Davie did with his very large research group over the years, and without the coagulation cascade in that work, I would not be standing here today. The quest of what we've done over the last couple of decades has really been to try to answer the question, can we turn genes into medicines? So, and whenever I say that, the CEO of Spark, which spun out from our unit at CHOP, always says no. The question is, can we turn genes into a business? So that crystallizes the dialectic that's going on there. All right, this is my forward-looking statement. And so let me just say a few words about gene therapy. The goal of gene therapy for genetic disease is really twofold. One is to get long-term expression of the donated gene, and the other is to express it at levels that are high enough to have a therapeutic effect. And what we do to treat hemophilia now can be summarized in this diagram, and essentially everything that we do involves recurring medical interventions, and after factor infusion, the factor level goes up, and then over time it declines, and then the patient's infused again, and it goes up. But it results not only in recurring medical interventions, but also in peaks and troughs, and during the troughs, people are at risk for bleeding complications. And so the goal of gene therapy for hemophilia is to maintain some level of clotting factor continuously so that we avoid these peaks and troughs, and we also avoid this necessity for recurring medical interventions. So where is gene therapy as a class of therapeutics right now? Well, the first gene therapies for genetic disease have both been licensed in Europe, not the United States. The very first one was licensed in 2012, and it was for a rare lipid disorder, lipoprotein lipase deficiency. But recently the sponsor announced that they would not continue with the post-marketing commitments required. It has not been a commercial success. So the other was licensed in 2016 by GlaxoSmithKline, and that is for a rare form of hereditary immunodeficiency due to adenosine deaminase deficiency. So that's licensed in Europe, but not the United States. And here in the United States, Spark has submitted to the FDA the application for the licensing of an AAV gene therapy product for a rare inherited blinding disorder, and that's also been submitted in Europe. So the dates for opinion on that will be in 2018. If it does go to licensing, it would be the first AAV product licensed in the United States, the first form of therapy for an inherited retinal dystrophy. And if it gets licensed in both the US and the EU, then that would be the first thing that was licensed on both sides of the Atlantic in terms of a gene therapy. So many of you know that hemophilia has long been a favored target of people interested in gene therapy, and there are a number of reasons for that. Gene therapy for hemophilia has the advantage of having a very wide therapeutic window. If you give the person at least 5% levels, you convert their severe hemophilia to mild, and if they go up to 100, they're still within normal limits. We don't need tissue-specific expression of the transgene, and many people in this room are very well aware of the fact that the recombinant clotting factors that we use to treat hemophilia now are made in Chinese hamster ovary cells and baby hamster kidney cells and so forth. There are genetically engineered mice and naturally occurring dog models of hemophilia, and those have been very important for many classes of therapeutics, but particularly for gene therapy in terms of allowing us to work out proof of concept. And finally, of course, thanks to the recombinant and earlier plasma-derived protein products, the endpoints in developing therapy for hemophilia are very well understood and easy to measure. And so, beginning in the late 1990s, there were a number of different strategies pursued to look at gene therapy for hemophilia A and hemophilia B. People used vectors ranging from plasmids to retroviruses to adenoviral vectors and AAV. And most of these were safe, but they were not successful in those two goals of long-term expression at levels high enough to be therapeutic. And the most encouraging data were actually from the use of an adeno-associated viral vector expressing factor IX delivered into the liver. And so where we find ourselves now in what I would call the second generation of AAV gene therapy trials for hemophilia is that there has been a clear coalescence of all efforts around the use of adeno-associated viral vectors. This lists a number of trials for hemophilia B, all using AAV vectors, some differences in the factor IX construct, some differences in the choice of AAV vector, but essentially they're all using AAV. And here you see a similar number of trials for hemophilia A. All the work in hemophilia with AAV began with factor IX because the cDNA is smaller and easily fits into the packaging capacity of AAV, whereas for hemophilia A, more engineering was required to get the B domain deleted factor VIII cDNA to fit into an AAV vector along with all the other required elements. So just to say a little bit about the recombinant AAV virion, I mean, in some ways this is, I think, one of the most complex classes of therapeutics that people have yet tried to develop. The recombinant virion has a protein capsid and then inside the therapeutic, the active agent, which is the DNA. And in this case, this is under the control of a liver-specific promoter, and this is the factor IX gene with a synthetic intron to expedite movement of the spliceosome from the nucleus to the cytosol. So the capsid consists of 3 different viral proteins, 1, 2, and 3, assembled in the ratio shown here, mostly VP3, 48 molecules of VP3 and 6 each of VP1 and VP2, assembled into this icosahedral structure. And to make the recombinant virion, the viral DNA from which this vector is engineered is fully deleted and replaced with the DNA that we wish to express. So the final recombinant virion is about 74% protein by molecular weight and 26% DNA. As I mentioned before, one of the few limitations of AAV vectors is that they will not package more than about 5 kilobases, and so fortunately most cDNAs in the human genome are shorter than that, but not everything is. So in our early work with recombinant AAV, we were able to show that introduction of it into the liver in hemophilic mice and hemophilic dogs resulted in long-term expression at therapeutic levels. And so with great confidence, we began the first trial in 2001. And this is a scene from the interventional radiology suite at the Children's Hospital of Philadelphia. It was a dose escalation study, and in those days we infused the vector actually into the hepatic artery under interventional radiology guidance. And these doses were taken based on studies in the hemophilia B dog model. And so we approached this thinking that it would be straightforward if we could if we could cure hemophilia in the hemophilic dog, which weighed 20 kilograms, surely we could scale it up to the 70 kilogram man. But in that early work, we learned the lesson that all drug developers know, which is that animals correctly predict the course of events in humans about 70% of the time. And this unfortunately did not fall into that 70%. And what we saw is that after vector infusion, the patient's factor IX level rose steadily to around 12%, and it stayed there for about 4 weeks, and then it began to fall, and it fell gradually back down to the baseline. And we had never seen this in mice, rats, rabbits, hemophilic dogs, or non-human primates. And this fall in the factor IX level was accompanied by a rise in the liver enzymes, which was asymptomatic and self-limited. So in other words, after a few weeks, it began to resolve on its own, and the liver enzymes returned to normal in tandem with the loss of factor IX. And it took us, I would say, a number of years in the laboratory to figure out exactly what was going on, but we eventually determined that human subjects who received AAV in the liver mounted an immune response to the AAV capsid. Now, interestingly, this same response does not occur if you put AAV into the subretinal space or if you put it into intramuscular injection. But if you put it into the liver, what we eventually worked out was that the breakdown of the capsid in the hepatocyte does eventually result in the presentation of capsid-derived peptides on the surface of the transduced hepatocyte, and that results eventually in detection by the immune system, destruction of the transduced hepatocytes, resulting in this rise in the liver enzymes, and simultaneously the loss of expression of the donated gene. But we did predict, based on this initial work, that short-term immunosuppression might be able to block that. And so when our friendly competitors at University College London mounted a trial beginning in 2010, as they went through dose escalation, when they got to the same dose where we saw this, they also saw a drop in the factor IX level and a rise in the enzyme, in the liver enzymes, but were able to initiate a course of prednisone. When this happened, we studied the lymphocytes from these patients and showed that in tandem with this rise in the liver enzymes, you see a marked expansion of a population of capsid-specific T cells in the peripheral blood, and that when you start prednisone, these are rapidly extinguished. And they were able to preserve some level of expression here in the range of 2% in these people. Now, eventually, in their study, 4 out of the 6 subjects who were dosed at this level required this course of steroids. But these results were extremely important for the field because they showed that you could get long-term expression of a donated factor IX gene introduced into the liver. This initial publication showed stability for 3.2 years after the initial infusion of vector. And a recent update on that shows expression out to 7.5 years from that first infusion. And by the way, progress since those initial clinical trials now allows us to infuse the vector intravenously, so that makes it a lot easier. So what we were interested in doing in our most recent work was trying to determine whether we could build a vector so efficient that it would allow us to drop the dose. These immune responses that require steroid management are a dose-dependent phenomenon. So we wanted to try to build a more efficient vector that we could give at lower doses and then see if we could get therapeutic levels of factor IX expression without triggering that capsid-directed immune response. And so we capitalized on a naturally occurring variant with high specific activity. So let me just say a little bit about what we are looking for in gene therapy for hemophilia. Obviously, we need long-term expression. And data from natural history studies in hemophilia suggests that, so this is factor VIII activity on the x-axis and the annual number of joint bleeds on the y-axis. And what you can see is that Obviously, if you move from less than 1% factor activity to the range of 5%, then the number of bleeds goes down very markedly. And if you can get up to around 12%, most people don't bleed no matter how they're managed. So that provides a sort of benchmark for where we'd like to get to. Obviously, you want consistent results so that every person who signs up for it gets a factor level within some known range. And as I said, obviously, we were looking to try to reduce the frequency of these immune responses, not because they're really a safety issue, but they are an efficacy issue. If you don't catch them and start the patient on steroids, they may lose all their expression. So, as I said, we capitalized on a naturally occurring factor IX variant, factor IX Padua, that was originally described in 2009 by Valter Arruda at the Children's Hospital and Paolo Simeoni in Padua. And this patient presented at a young age with a thromboembolic event and was found to have a single mutation in the factor IX gene, arginine 338 leucine, that conferred an increase in specific activity of the factor IX in the range of 3 to 8-fold. We used a capsid that had strong tropism for liver in studies in animal models, and we used a strong liver-specific promoter in the recombinant virion. The structure of the trial is shown here. This initial study was for males at least 18 years of age with factor IX levels of less than or equal to 2%. Very important point here, they could not have any appreciable circulating neutralizing antibody to the AAV capsid that we were using. Because AAV virions are derived from a naturally occurring virus, a substantial proportion of the adult population carries antibodies. They were exposed during childhood, probably through a respiratory tract infection. And if those titers are high enough, we can't get past those. A lot of the effort in the field right now is directed to how are we going to overcome these pre-existing neutralizing antibodies. But right now, the way all the trials deal with it is that they screen out individuals who have any appreciable titer of neutralizing antibody to AAV. They had to have previously received treatment with factor IX, and they could have a history of Hep B or Hep C, but they had to have undergone clearance of HCV with direct-acting antivirals and not be left with any substantial level of liver fibrosis. Again, if HIV positive, they needed to be controlled on antiretrovirals. And these were the other conditions, baseline treatment, either on prophylaxis or on demand with some documented bleeding episodes. We initially structured this as a dose escalation study, but as you will see, the results at the lowest dose were such that we did not dose escalate. So what this involves for patients who meet all the eligibility criteria is coming into the clinical trials unit, getting a 1-hour intravenous infusion of the vector. They stay there for a few hours to be monitored, and then they go home and have blood work checked frequently through the first 12 weeks. Typically, if people have an uneventful course over the first 12 weeks, then they're just in long-term follow-up. We are currently working on this program at Spark in collaboration with Pfizer, who will be taking it forward into phase 3. So this shows some data on the first 10 subjects. They were all men with severe or moderately severe hemophilia. Most of them had less than 1% baseline factor IX. A few of them had levels between 1 and 2%. They ranged in age from 18 to 53. Most of them had no pre-existing antibody to AAV, so less than 1-to-1 titer. We had one person in here who had a low titer antibody. As you would expect for a group of patients with factor IX deficiency, everybody had a different mutation except for two brothers here. Most people were on prophylaxis at this point. A few of them were on demand. And a few patients who had done a good job with their prophylaxis through the years had no target joints, but then other people, particularly older people, did have target joints. Many people had been exposed to Hep C. So this shows the table of adverse events in these initial studies, and most of these are routine things that occur in patients if you follow them over a year. There was one event that was likely related to the study agent, and that was transaminase elevation in one of these people, and I'll show you that. But most of the patients pursued a course like this very first patient. So this was a patient who was infused in December of 2015, right after ASH. And you can see that his factor IX level slowly rises over a period of about 12 weeks to a plateau of around 30%. And this shows his liver function test at the same time, and you can see they edge up a little bit within the normal range after the vector infusion, but they never get out of the normal range. And then they pretty much remained the same over the following. This shows data out to about 18 months. Since the patient was infused, he was able to stop all of his prophylaxis. He never needed any course of steroids. He has not had any bleeding, and he has not used any factor. In fact, this is a patient in the United States, as you may guess. After about 4 months, his insurance company called to make sure he was still alive because he had not used any factor for 4 months. And so of those other 10 subjects, a total of 8 of them had a course very much like what I just showed you. So over a period of about the first 12 weeks, you follow them carefully, and they reach their plateau level, and then that's pretty much what they've done in the time period since then. Among these people, the mean steady state factor IX level was about 30%, and there was nothing in the liver function test that caused us to need to administer steroids. And you can look at the individual curves of all these 8 people, and you see that they all look pretty similar. So I'll just point out that there were also 2 subjects in whom we did detect an immune response, and this was the first of those. He was the 7th person infused, and you can see that his factor IX level initially rises quickly to about 50% of normal, and then it falls, but you see he had had a kind of seesawing effect anyways, so we did not immediately respond to this by starting steroids, and that turned out to be, not less than ideal. And but eventually when he fell to the level of around 20%, we did understand that you see this rise in the liver enzymes is really pretty subtle at this dose. Because the dose is low, you don't get much of an effect even when you're getting an immune response. But the LE spots tell the story. These are, again, measuring capsid-specific T cells in the peripheral blood, and you can see that right around 6 weeks, there's a marked expansion in that population of cells, and that correlates with this very mild rise in the transaminases and the drop in factor IX. So the patient was begun on prednisone, and it was tapered slowly over a long period of time, and he stabilized, his factor IX stabilized at about 16%. So actually for him, that's been a good outcome. He has not needed factor infusion and not had bleeding episodes at that level. And we had a second subject who had a similar course. This one was much easier to spot because there was a marked rise in the liver transaminases at the same time. And the patient was begun on steroids, and he actually stabilized at around 79%. These LE spots take about a week to come back, so you have to really, they can confirm your impression, but you often have to act without them. Just to call out a couple of other interesting aspects of the data, among these first 10 subjects, 6 of them were missense mutations, and 4 had stop codons or frame shift mutations. So all the missense mutations were CRIM positive, and all of these others were CRIM negative. If we take out the 2 subjects with the immune responses and the person who had a low titer neutralizing antibody to begin with, and then look at the others to average the levels, you can see that the CRIM positive subjects have a higher circulating activity level than the CRIM negatives. And I think that that's consistent with current concepts of the large extravascular reservoir of factor IX, which in the CRIM negative patients has to be filled. So what are the confidence intervals for dosing? I'll just show you a little data here. So this is all the 10 subjects put onto the same graph. And what you can see here are, the two, here's the subject in blue, light blue, that we didn't catch the immune response really as early as we might have. And so he fell down to this level of 16%. This person at 18% was the person who had the low titer pretreatment neutralizing antibody to AAV. And now we have as a control in the study his brother, who's shown here in green. So same mutation, and he gets the level of, but no pre-existing neutralizing antibody to AAV, and he gets a better level. So are those related? Well, we need more experience to figure that out, but I suspect they are. And so if we remove the people with the neutralizing antibody and the immune responses that we didn't catch, you see that the levels of the subjects range from 24% to 79%. I suspect that we will not be able to get better than about a threefold window. And if you look at all the steps that are involved in AAV transduction of a hepatocyte, you have to get the AAV to the hepatocyte, bind to the cell surface receptors. The vector is internalized in an endosome, and then it has to escape from the endosome. It has to uncoat and go into the nucleus and make the second strand. I think when you look at all those steps, it may be difficult to get better than about a threefold target for the confidence interval. So it matters, I think, what you aim for. So this just shows that the annualized bleeding rate has been markedly reduced in all of these individuals. This shows the number of bleeds that they had in the year prior to vector infusion and then in the time course since. And so 9 out of these first 10 subjects had no bleeds in the year after they got vector. One person had 4 suspected bleeds. This was one of the people with a lot of arthropathy from his hemophilic disease, and these were suspected bleeds, not confirmed, but he treated himself for them. And then this compares the number of infusions that all of these people underwent in the year prior to vector infusion, and then the number of infusions in the year since. So let me just say that the areas where the field is working very hard right now, what will be the durability of expression? I mean, we know from published data that there are at least 3 years of expression in those initial subjects from the trial in London that began in 2010. We know that we have over a decade of expression in hemophilic dogs from a single injection. An area of great interest right now in the field is whether it is possible when people are injected with the AAV vectors to block the formation of neutralizing antibodies so that they can be re-administered later on if expression does begin to wane. And then, of course, I mentioned that there's a lot of interest in trying to develop strategies, devise strategies more sophisticated than, let's say, plasmapheresis to overcome the pre-existing neutralizing antibodies to AAV that exist in 30 to 40% of the adult population. As I showed you in that earlier slide, there's a lot of interest now in extending to hemophilia A. Promising results are beginning to be reported. There'll be more at ASH. And eventually, of course, people would like to extend to the pediatric population. That will almost certainly require traversing this issue of re-administration. So I'm actually just going to conclude now by thanking all of my colleagues at Spark Therapeutics. Many of them were people that we worked with initially at Children's Hospital of Philadelphia, and they moved over to Spark with us. Our clinical trial sites have been led by Dr. Lindsay George at the Children's Hospital of Philadelphia, and so these are some of the people on her team there. Other clinical trial sites for us have been the University of Mississippi, University of California at Davis, a terrific site at St. Michael's Hospital in Toronto, Royal Prince Alfred Hospital in Sydney, Australia, and Pittsburgh and Cornell, as well as our collaborators at Pfizer. So thank you very much for your attention, and I am happy to take questions.
So we have a mic runner, though. Mic runners. Please say who you are.
Cedric Carter, PBR. The only thing I didn't get, I hope, was the CREM positive and CREM negativity. Why would that make a difference?
Well, I think that it has to do with the fact that the volume of distribution for factor IX, so in the CREM negative subjects, the material that's coming from the donated gene has to be distributed into the extravascular space.
I'm with you now.
In the CREM positive subjects, that's presumably already filled up.
Occupied, I'm with you, yes. Okay, I'm with you, thanks.
Okay.
Yeah.
Yes.
Sorry.
Well, I can hear you. I'll repeat the question.
Thank you. Just a question. Have you considered giving prednisone all? You had that one where
Yeah.
there was the 79%, and you said in one of the patients you missed it, the elevation in the enzymes was very subtle. You think giving a short course of prednisone to every patient might make a difference?
Well, I think that's a really important question for the phase 3 study. You know, in phase 1-2, we try not to do that so that we can try to figure out at this dose what is the frequency with which we are going to see this. So out of these first 10 subjects, 2 people needed steroids. Would you give them to everybody for the 2 who needed them? And you know, I think that's just a clinical judgment question. It's a really important question, but it's good to do the initial studies without it so that, you know, because each vector and each dose of vector has a signature at about when the immune response will occur if it occurs. So the window here looks to be at about 5 to 6 weeks. So you have to do it without steroids first to know when it is, so you know when to give it. But you know, that's an area that's being actively discussed for the phase 3 trial. And you know, I mean, the reason is exactly as you surmise. There's a heavy penalty for missing it. So, yes, yeah.
Really nice work. I'm wondering, so with the levels that you're achieving in factor IX, it's because of the mutation factor IX
Right.
PADWA. So what do you think would be the strategy for factor VIII? Because it really, I mean, I think it's fortuitous that you have that kind of an expression mutation. So what would,
Right.
are you
So,
seeking
yeah.
ways to imitate that for factor VIII?
Well, they're short of making a somehow better cofactor, and I don't know if that's possible. The strategies that people are actually pursuing is either you can use a more efficient capsid, or you got to dial up the dose. If you dial up the dose, everybody's going to have an immune response, and you got to be sure that you can manage that. So, you know, again, if you go on the steroids, you know, it's a course of anywhere from 6 to 12 weeks of gradually tapering steroid doses. I don't think that is, most people would not have a contraindication to a 6 to 12 week course of steroids, so just going up on the dose may be one successful strategy for factor VIII. And that's basically what another sponsor, BioMarin, has done. And we're pursuing a strategy of using a more efficient AAV capsid. So, you know, these are the kinds of things that will play out in clinical investigation. Yes.
Thank you. That was great. I have a question also related to hemophilia A, obviously a larger population worldwide of patients. and specifically how the continuous preservation of a factor level might interact with the patient's own, pharmacokinetics, kind of like with the factor 9 when you have CRM positive and negative and all these extravascular things to consider. Obviously with factor 8 we're primarily intravascular, but we have von Willebrand's factor, and I'm sort of wondering, and I've always wanted to ask this question about what your thoughts are, or have you seen any difference in patients who have sometimes a 3 or 4-fold variation in their baseline von Willebrand factor activity, and is that something that could be considered as a way of minimizing dose, because their von Willebrand's is going to be able to help that factor 8 be more preserved or at a higher level?
So we have wondered about that for some of the things that we've seen. I would say right now, the total experience in the world with successful administration of AAV factor 8 is too modest for there to be enough patients to look at that sort of thing. So no answers yet.
I'm
Well,
assuming
people are
you're
interested
following
in
it.
that, yeah.
You're following the von Willebrand
Yes.
and blood group and all that kind of thing.
Yes,
And the
blood group and all that.
Yeah. And the age of the patient, because clearly there's variation.
Yeah, now, we're not using the native promoter. So things that are true for normal factor 8 expression, acute phase response, all that sort of thing, are not going to be true for the donated gene. So, anyway. Yes.
So I have a question on the hemophilia A therapy as well. So, factor A is a very large gene, so BioMarin actually have to use a very large dose to treat the hemophilia A patient. And I know Sparks is also pursuing the clinical trial. So were you able to make sufficient titers for AAV factor 8?
The BioMarin doses are very high, I think. So sorry, this is AAV geeky talk here. But they make their vector in insect cells, in a baculovirus system. And what we've seen in all the hemophilia trials is that vector made in baculo needs a much higher dose than vector made in mammalian cells. I think that the reason for that is that, AAV, the natural host is humans. So when you make AAV in mammalian cells, we make it in HEK293 cells, you get the right ratio of VP1, 2, and 3. The challenge of making it in insect cells has always been that the ratios of VP1, 2, and 3 are not the same because the codon usage is different. And so people have really tried hard to get the ratios to come out right, and I think that's been a challenge. And when you don't have the right ratio of VP1, 2, and 3 in the recombinant virion, you get a defective particle. So it packages DNA and you count it on PCR titering, but it doesn't bind to the cell surface receptors and internalize and escape from the endosome in the same way. So those doses always look much higher, but I think the effective dose is probably similar. Sorry, that is AAV geek. Okay.
Question. So inspiring and exciting. Kathy, I'm wondering once
And it only took 25 years.
No kidding. We're on an exponential phase now. Can you give us a little light on, after we know a little bit more about what's happening in adults, whether it makes some good sense to start transfecting infants who are hypoimmune?
Well, I think that the real clinical development pathway will probably go initially to adolescents. One of the benefits of AAV in terms of safety is that it does not integrate. But that means that if you go into a child whose liver is going to grow a lot, you will lose your levels, right? Because you're not passing it on to the daughter cells, it's not integrated. So we're going to have to be successful at this problem of overcoming neutralizing antibodies to AAV, because as soon as you give the child their first dose, their AAV titer is going to go very, very high, and then you're not going to be able to dose again. So we're going to have to figure out how to block the formation of neutralizing antibodies, and there's a lot of work going on that question. So I think what you'll really see is it'll be worked out for adults, then people will go to adolescents, And then eventually, you'll begin to see the pediatric population come online.
So our next speaker is Carol Meow. Carol is a principal investigator at Seattle Children's Research Institute and a professor of pediatrics at the University of Washington. She has an interest in hemophilia and gene therapy in particular. She was, as I understand, one of the very first to introduce non-viral naked DNA approaches in hemophilia A and B mice. And so we have the pleasure of hearing her present her talk on novel approaches to treat hemophilia. Carol.
Okay. First, I would like to thank Ed Conway, Ross McGarvery, and the organizing committee to invite me to give a talk today. It is my great honor to speak at Earl Davie Symposium, especially because I was previously trained in Earl's lab. So I have nothing to disclose. Sorry. My name tag. Okay, so before I tell you our recent work, I would like to give my special appreciation to Earl. If I had made any slight contribution to the science in coagulation, it should be attributed to Earl, who gave me a chance to start my enthusiastic journey in this field. How do I describe Earl's lab? If you allow me to quote a Chinese saying, it is a place where tigers reside and dragons hide. Of course, Earl is the greatest and fiercest dragon. I remember Aki Ichinose in the lab used to call him Hancho, which means big chief in Japanese. And Jose Lopez, correct me if I'm wrong, called him Earl. Whereas I was scared, so I always called him Dr. Davie. Of course, I'm kidding because you all know that Earl is the perfect and nicest gentleman. And I learned to be a responsible scientist from him in many ways. In his lab, I worked with Kazuo Fujikawa to learn protein chemistry. And later, I was also mentored by Dominic Chung in molecular cloning and genetics. I felt I was prepared to pursue research in gene therapy for hemophilia when I went down to the gene therapy field. So this slide shows the coagulation cascade proposed by Earl in 1995, which demonstrates the importance of both intrinsic and extrinsic pathways that constitute the whole picture of the coagulation system. So in recent years, my lab focuses on research on improving the treatment of hemophilia A arising from factor A deficiency. So Kathy actually already gave a pretty good introduction to hemophilia. So I just want to say that the major improvements that needed for hemophilia treatment because of the current therapy involves repeated and infusion of bacterial recombinant proteins. So it is needed improved treatment for effective and safe and less costly treatment. And particularly, we focused on developing gene therapy for hemophilia to achieve persistent expression of factor VIII. And secondly, many patients, as you all know, that develop inhibitory antibodies against factor VIII. So we try to develop immunomodulation and cell therapies to overcome this problem. So I also have to tell you that I was worried when I learned I have to give a talk after Kathy Hai, because it always seems that she has solved most of the problems. But anyways, in my lab, we believe that there are many ways we can improve hemophilia gene therapy. And so my lab has focused on developing improved therapies for hemophilia treatment, as I just mentioned. So we have, over the years, we have developed a safe and efficient non-viral gene delivery method, in particular, ultrasound-mediated gene delivery of naked plasmid, naked factor VIII plasmid, to which we demonstrated that using this UMGD method, we can achieve phenotypic correction in hemophilia A mice. And this work has proceeded to large animal studies currently. And secondly, we try to develop immunosuppressive therapies and also in vivo regulatory cell expansion, and most recently Treg cell therapy, to try to prevent the anti-factor VIII antibody production. And today I would like to focus on and tell you one of our most recent hemophilia A gene therapy methods by using intraosseous delivery of a lenti factor VIII vector into the bone marrow. So why, if the AAV already showed a significant promise to treat hemophilia, so why choose lentivectors? I actually missing a slide here. So I would just tell you the reason why we chose lentiviral vectors. Firstly, as Kathy also already mentioned, that AAV persists as episomal vectors after in vivo gene delivery. So over time, transgene expression may decrease, and repeated dosing will be required. And it remains to be seen. I think there's still quite, a lot of people are investigating this, but it's still at this time whether this is plausible is still a question. Whereas lentiviral vectors is an integrating vector. So if successful, a single treatment of lentiviral vector-mediated gene therapy will be sufficient for a lifelong effect. Secondly, lentiviral vectors can accommodate large genes, more easily accommodate very large genes such as factor VIII cDNA. And additionally, lentiviral vectors can efficiently transduce both dividing and non-dividing cells, leading to significantly increased efficiency targeting primitive stem cells compared to AAV and traditional retroviral vectors. And lastly, recent clinical trials of ex vivo lentiviral gene therapy for several primary immunodeficiency diseases showed significant clinical benefit without adverse effects or aberrant clonal expansion. Therefore, my lab has developed this new method by using intraosseous delivery of factor VIII lentiviral vector for hemophilia gene therapy. So recently it has been shown that intraosseous delivery of several vectors can transduce liver cells, hematopoietic stem cells quite efficiently. And my lab has devised a more, try to improve the transduction efficiency by slowly infuse the vector intraosseously into mice using very low speed and very small volume, such as 20 microliters with infusion speed about 10 microliter per minute, which is precisely controlled by a syringe probe. In this way, we found that hematopoietic stem cells can be very efficiently transduced by the lentiviral vectors. And furthermore, this method actually can bypass many obstacles encountered by ex vivo stem cell gene therapy, which in particularly ex vivo therapy requires preconditioning, which is and requires some potentially like irradiation or some potentially toxic agents, which can induce serious, severe thrombocytopenia, and that is highly undesirable for a hemophilia patient. Therefore, I just want to show you our mouse data. So when we inject these lentiviral vector encoding GFP driven by a ubiquitous promoter, MND promoter, on day 7, we actually can see that we achieved up to 15% GFP expression in the total bone marrow cells, but most significantly, we can transduce HSCs very efficiently, up to 17%. So these data actually show that a single IO injection, that's intraosseous injection, can lead to very efficient transduction of the HSCs in the bone marrow. And with this success, we made a factor VIII lentiviral vector driven by a ubiquitous EF1 alpha promoter, and we used the high titer. We used the high titer lentivirus and intraosseously deliver this vector into the mice. And as you can see, we obtained initially very good levels of factor expression, up to about 10 to 20%. Unfortunately, the level drops very quickly, and it coincides with the formation of the inhibitory antibodies. So that's anti-factor VIII inhibitory antibodies. And we think that the reason for this is because after vector infusion, because this vector was driven by the ubiquitous promoter, so it can express in many different cell types, including antigen-specific cells, which may have enhanced the induction of inhibitory antibodies. So with that, we actually we next actually adopted a strategy of targeting the factor expression in platelets to avoid the antibody, avoid the neutralization of factor VIII by antibodies. So this concept was not developed by us. I mean, it was initially developed in transgenic mice and ex vivo gene therapy by a couple groups, Morty Pons at UPenn, and Bob Montgomery and Chi-Jian Shi at Milwaukee Blood Center. So why do we want to target factor expression in platelets? So in our case, once we deliver the lentivector into the hematopoietic stem cells, then using a vector that's driven platelet-specific expression, then once the stem cell differentiate into megakaryocyte, then factor VIII will start to express in megakaryocyte and eventually be stored in alpha granules in the platelets. And these factor VIII-containing platelets can then circulate into the blood. However, the factor VIII was protected from the neutralizing antibodies because it's not secreted in the blood, whereas it is contained in the platelets. Most significantly, once injury occurs, then the factor VIII can be released directly into the injury site without being influenced by the factor VIII antibodies. So using this strategy, we made a factor VIII lentiviral vector driven by the platelet-specific GP1B alpha promoter. And as you can see that factor VIII was not expressed in the HSCs, and it was not also found in T cells, B cells, and myeloid cells in the blood. But on day 91, we can see up to 3.3% of the factor VIII was expressed in the platelets. So that's figure C there. And with that, we looked at the percentage of platelet that contains factor VIII by flow cytometry, and we can see that about 1 to 3% of the factor VIII was maintained in the platelet from day 27 up to day 160, and that's the experimental duration. And to test the phenotypic correction by by factor VIII containing platelet, we did the tail clip assay in the treated hemophilia A mice, and as you can see on the right panel there, we achieved partial correction of the hemophilia A phenotype in the treated hemophilia A mice. We also asked the question whether IO infusion of this lentiviral factor VIII vector that specifically targeting expression in the platelets can also achieve phenotypic correction in hemophilia A inhibitor mice. So the inhibitor mice were generated by a repeated injection of human factor VIII protein many times. And but that we obtained about the average inhibitor titer was about 80 Bethesda units before the IO infusion. Then we infused the G factor VIII lentiviral vector into the mice via IO infusion. And as you can see from the left panel, we achieved about 0.74 milliunits per mil per 10 to the 8th platelets in the on day 27 in the treated mice. And then in the right panel, it also showed that even in the inhibitor mice, these factor VIII containing platelet can correct the hemophilia A phenotype in these inhibitor mice. So these data are very encouraging to show that one single treatment of platelet-specific factor VIII lentiviral vectors can offer phenotypic correction in hemophilia A mice either with or without inhibitory antibodies. So we continued in the lab, we continued to try to achieve even better effect. Hopefully we can use less vectors or can have a higher correction activity for the hemophilia. So in this slide, shows that this is the observation actually we found in the lab is that in immunodeficient mice, the factor VIII, well, this is a GFP experiment, but similar with factor VIII, these transgene expression levels actually maintain pretty high levels over time, whereas in immunocompetent mice, they tend to drop gradually over time. So in order to, so we think the reason is because there's probably innate immune response occurred after lentiviral vector infusion, and eventually there could be associated cytotoxic effect against the LV transduced cells. So in order to overcome this and to achieve higher transduction efficiency, we used a combination drug treatment with dexamethasone to overcome the innate immune response, and followed by the administration of anti-CD8 monoclonal antibody to decrease or eliminate the cytotoxicity potentially induced against LV transduced cells. So this slide shows that with a combination treatment, it can significantly, as you can see on day 7, even with dexamethasone, I think it's because the reduction of the innate immune response probably helped the increase of the GFP positive cells in the total bone marrow, and most importantly, over a longer time at day 160, we can achieve higher transgene expression in immunocompetent mice with drug treatment. And we also asked the question, I think this also reflects to, using a higher activity factor 8 compared to the regular factor 8 cDNA. So in this case, we adopted a recently developed factor 8 variant. This is a kind gift from Weidong Xiao at Temple University. So factor 8 X10K12 contains a deleted B domain. It has 10 amino acid change in A1 domain, and then 12 amino acid change in the light chain of the factor 8. So we actually first made the lentiviral construct. These are the plasmids, and then we just hydrodynamic injected these plasmids into mice to test the activity of these factor 8 molecules. And as you can see, that after hydrodynamic injection, the activity increased tremendously, almost 25-fold better than the factor N6 molecule that we usually put in our vector. So we were very excited. So we made these vectors driven by the platelet-specific GP1B alpha promoter, and then put them into mice via IO infusion. And as you can see in the tail clip assay, the vector with the factor variant and drugs showed improvement in correcting the phenotype. However, as you can see on the right panel there, although the mice treated with the G factor N6 vector or the factor X10K12 vector plus drugs had no inhibitor or low titer inhibitors, but the mice treated with the G factor X10K12 vector actually had induced very high titer inhibitor antibodies. And I think the reason is because X10 or the 10 amino acid change in the A1 domain actually increase significantly the secretory function of activity of factor 8. So probably quite a bit of factor 8 went into the circulation and induced the anti-factor 8 antibody. So to prevent that, we actually used the alternative. Next, we tried to use another variant, which is factor N6K12RH. So this variant contains a B domain deleted factor 8 and 12 amino acid change in the light chain, and a single amino arginine to his mutation in the at amino acid 1465. So with this variant, it is predicted that it would produce higher activity, higher functional activity, but without increasing the secretion of factor VIII. So as you can see from the hydrodynamic injection experiment, we obtained about threefold increase in factor VIII activity compared to the factor N6 molecule in our original vector. And we were very pleased to see that on day 64, we did not see any factor VIII activity or inhibitory titers in the plasma in these treated mice. However, we did see factor VIII expression in the platelets, which indicate that factor VIII was predominantly stored in the platelet and did not escape into the blood circulation. So on the right panel there, we actually evaluate the phenotype correction of these hemophilia A mice using the blood flow rate assay in a carotid artery injury model. I think this is a more accurate assay, even better than tail clip, to very objectively evaluate the phenotypic correction. And we're very pleased to see that with this variant, after combination with the drug treatment, significantly reduced the blood flow rate compared with our parent vector, G factor N6 lentiviral vector, and getting to be had a correction very close to the wild type mice. So what's next? Okay. So we think that, of course, the goal is to hopefully to push this new strategy into to benefit hemophilia patients in clinics. So in the lab, we try to test the transduction of these vectors in human CD34 cells and in humanized NSG mice. And we also just started in collaboration with Tim Nichols at UNC Chapel Hill, we just started our first hemophilia A dog experiment. And this is in use with the IO infusion using a pediatric bone injection gun device, and which can have easy access. I think I need to go a little bit faster, but anyway, so sorry, it's a little slow. But anyway, so with this device, we were able to gain a very rapid access in the dogs, and the whole infusion process can be completed in about 25 minutes. And I'm glad to report to you that the first dog tolerated the procedure quite well and has recovered very well. And this is using a lower titer as the first batch, lower titer lentiviral vectors. And this is, we continue to monitor the therapeutic effect. And also to look ahead to go to clinical hemophilia gene therapy, and there are these new intraosseous device that's FDA approved, can rapidly and easily gain access to either humerus or tibia within seconds. And we believe that the infusion of the vectors can be completed in very reasonable time to efficiently transduce the bone marrow cells. Okay, so this is just a conclusion that I talked about. So, but I think we're very hopeful that hopefully the IO infusion can efficiently correct the phenotype in large animal model, such as in hemophilia dogs, and can pave our way to human clinical trial. And lastly, I just like to acknowledge all the people in my lab who has done wonderful work, and Chong Li and Xiefeng Wang are the leaders of these projects. And I also like to acknowledge David Rollins and Iron Kim in Seattle Children's Research Institute, Tim Nichols at UNC, Morty Pons at UPenn, Weidong Xiao from Temple University, and Dao Pan at Cincinnati Children's Hospital. And I'd like to thank the support from NIH. Thank you very much for your attention.
So I'll start. Very nice talk, Carol. And thanks for also acknowledging our boss, Hancho. So I'm wondering about the strategy of targeting the factor VIII to platelets. It sounds good for keeping it away from antibiotics. What about its ability to stop bleeding in the joints?
Yes, that's a good question. We haven't really looked into that. So we think it should help, but we don't know exactly, because you're expert in platelet. What do you think? Would it help in the joint?
Well, yeah, I don't know. Definitely platelet and platelet products can get into joints and cause problems, as in rheumatoid arthritis and other things, but whether or not that happens when the joint is normal and whether you can get a secreted protein in there is a question, and I just think it's an interesting part of what you're doing.
Right,
Yeah.
right, yeah. And I also think the platelet therapy is, it kind of has many usages, I think. First thing is we like to use it not only just for, particular treating this particular population of hemophilia patients with inhibitory antibodies, because these patients really lack very ideal treatment for them. But also we can also think about using this therapy to induce tolerance for these patients. And another thing is, I think it could be, used in combination with other therapies as well. So like you said, maybe in the joint bleed, maybe it needs extra help, but it would not be nice to have some baselines there for these patients to start with.
Well, one thing I would say about that is that you should get some sense of that from the experiments in the hemophilia dogs, because they do get joint bleeds. So should have some indication.
Yes.
I had a question about the factor VIII X10K12 results that you showed on the model.
Yes.
It looked like, although it improved bleeding, it didn't come anywhere close to the wild type reduction on the tail clip assay.
Right.
Do you have other data that speak to the activity of the factor VIII variant?
Yes, so, in the hydrodynamic delivery, we get very, very good activity compared to the factor VIII, the B domain deleted, BDD factor VIII. But I think it's a little bit different when it goes into the storage in the platelets compared to, that's why we get great expression, but then it also went into the circulation and induced antibodies. That's why we try to shy away from the X10 variant and just use the K12. So if you look at the my last slide with the K12 only.
Yeah, I saw that your results were better with that carotid artery blood flow model.
Yes,
but
yes.
why wouldn't you see the same thing
In the tail clip.
In the tail clip.
Right. So somehow I think in tail clip, because it's partial correction, and in tail clip, also I think the platelet factor VIII, this we have to go back and check, talk to Morty too, but I think the platelet factor 8 is a little different than the plasma factor 8. That's why it still needs some combination therapies together with that. It's a good baseline. That's what I would think, yeah. But we can discuss that some more. I think it's very interesting, yeah.
Thank you very much. That works. Okay, but our next speaker, I'm not going to really do much of an introduction. So this is just George Stevenson. He's not a scientist, as far as I know. No, George? And he's not a physician. He's invited as a special guest to tell a story. And that's all I know about him, other than he's a Canadian.
Before I start, I want to say directly to Dr. Hai that I did not expect the emotional content to be quite as high as you made it this morning. And as a hemophiliac, I want to express to you, on behalf of all hemophiliacs, the inexpressible gratitude that we have for the work that you and your colleagues are doing. So, ladies and gentlemen, my name is George Stevenson, and I have mild hemophilia. I'm 84 years old with a bow to Dr. Davie, and I came into the world with mild hemophilia, and I probably will go out of the world with that intact. I have a nephew who is about 60, and I have a 10-year-old grandson with mild hemophilia. So I'm not a medical expert, but I have what you might call a significant amount of practical experience. Mild hemophilia is very different from moderate and severe hemophilia. It's so different that it might be described in a different way. It might be viewed as a different kind of disorder, except we know that the physiological stuff is the same for all of us. Unfortunately, the word mild frequently leads to an assumption that the condition should not be taken seriously, doesn't have to be taken seriously, that it's not dangerous and it doesn't need attention. And I can tell you, with a lot of experience, that that is absolutely not true. The main characteristic of mild hemophilia is, unlike the moderate and severe forms, it's highly unpredictable. In fact, it's downright sneaky. 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 And while being sneaky, it can get away from us when an event occurs, so that it can get bad, it can get painful in the extreme, and very hard to fix. So what I mean by sneaky is a few things like this. If one of us, as a teenager or young person, falls or has an accident or gets bumped, we know what's going to happen if we are moderate or severe. But with mild hemophilia, we don't know. Sometimes it goes on to a bleed, and sometimes it doesn't. We can wait to see what occurs and get lucky, or we can wait to see what occurs with disastrous results. A bump or a fall can take up to 2 or more days to manifest itself. Then, if we haven't caught it, we're in big trouble because it hasn't been treated with factor in the intervening period. In a manner of speaking, it picks up steam and gets away. That is why the instructions of all hemophilia clinics is treat when in doubt. And I can tell you with certainty that is the absolutely best advice around. Some events in my own life that have illustrated this. A fall on ice when I was 16 that put me in hospital and then in bed for months. A car door slammed a little bit too hard on my hand when my children were young and growing up. Bumping my head on an overhead pipe while I was walking through a garage on my way home singing a song. Twisting an ankle at the edge of a sidewalk. Falling on black ice in a cement stairway. Biting my tongue while enjoying the eating of fresh unpitted summer cherries. It bled for a very long time. Going to a dentist for remedial work and waking up 2 mornings later with one of those disgusting clots in the making filling my mouth. These sneaky situations have remained for me all of my life. And while age means less trauma from sports and from the things you do when you're young, that's replaced as one ages with things like these. A vertigo-induced collapse in an elevator when I broke both a tooth and my nose and I did substantial damage to my face. Open heart surgery and a host of related cardiac procedures, including angiograms, a bypass, stents, an artificial aortic valve, and subsequently a valve-on-valve. The repair of a hernia, a bleed into a knee after an extravagant evening of dancing, a TERP, where bleeding kept on recurring for weeks and weeks and weeks until I transferred to a hospital where there was a hemophilia clinic. And the increased danger of tumbles and falls as mobility, eyesight, and general steadiness decreases. The sneakiness of the condition, mild hemophilia, prevails throughout a lifetime and should not be ignored. It's truly troubling that many in the medical and dental communities are unaware of what hemophilia generally and mild hemophilia in particular means. For me and others I know, friends, that has meant we have to be our own advocates, frequently needing to be much more outspoken than I would want to be. This applies to most medical personnel, both in and out of hospital. Within hospital, I can count several times that I have told emergency and other staff that contact with my hemophilia clinic is essential, and even then it was disregarded. If I had not been vigilant and eventually demanding, and if I had not been friends with my friends in the hemophilia clinic, I probably wouldn't be alive today. I particularly want to say, in addition, that a long time ago I lost count of the number of times that I've been told by a dentist and others not to worry, this is just a little poke, and bleeding is very rare, only to pay the consequences of a bleed which is very difficult to control. Many years ago, I learned that language, and I learned to say, stop. I have to have factor. I have to have factor. For those following me, for my grandson, my wish is that awareness is raised so that we don't have to rely so much on our own resources. Please remember that mild hemophilia, while not terrifying is sneaky, tricky, and dangerous. With appropriate input from hemophilia clinics, that can end. Thank you very much for letting me make this presentation today.
Are there any questions or comments? Shannon, do you want to say something? Do you have a mic? We have a mic? Our runners are coming. Run! That's why you're called mic runners.
George, thank you so much for that you can portray a lifetime of interaction and cycling of hemophilia care better than any medical professional on the planet, I think. And I just wanted to say to the audience, you know, I think it's people like you who teach us as clinicians how to move forward. And it was actually George who coined a term that I use in almost every visit with a mild hemophilia patient, is that it's sneaky. And that's a huge lesson that you'll never see in a textbook. You'll never read about it or you'll never see it at a conference presented as such. But I think that's the reality. And thank you for presenting it so incredibly well to our combined scientific and medical audience. And we have a lot of people who will take away something from your presentation, so thank you. And I just wanted to say we have a case that we have next, which I think you've perfectly set the stage for in terms of one descriptor of how things can go in sneaky hemophilia.
Thank you, Shannon. My pleasure.
Well, I wonder if you'd be willing to share with us what your factor level is.
Well, my factor level is somewhere between 12 and 14, so I'm, you know, amongst others, kind of blessed. But, you know, when you were making your presentation, I was thinking, well, we're eventually going to get to mild hemophiliacs.
Well, you know, I think when it does get to mild hemophilia, there'll be lots and lots of safety data from severe and moderately severe. But I think, you know, right now, I don't think the regulators would look with favor on including people with mild hemophilia. And it may be that part of the reason for that is that they don't understand what you just spent 10 minutes explaining.
I have a feeling that's so. My grandson's level is between 5 and 7. Thank you very much.
Well, Shannon has already introduced our next speaker. All right, whoops. Drew Bowie is a medical resident, a first-year medical resident. I guess has rotated through the hemophilia program a little bit, or not. By proxy, okay. Anyway, he'll tell you. But he is, as I said, a first-year medical resident. He has a background, a Bachelor of Science, and he has some actually considerable basic science background as well. And he's agreed to give a little talk about the sneaky aspects of mild hemophilia. Sure.
Hi, everybody. Thanks again to Dr. Jackson for inviting me to do this talk. This is, you guys might be sensing a theme with the sneaky aspect of hemophilia here. This is a case that I saw when I was actually a medical student working in the internal medicine unit at Royal Jubilee Hospital in Victoria. So just a little bit of background here. Mr. S, when I initially saw him, was a 76-year-old gentleman, but his history with hemophilia kind of goes back much longer than that. And when he was 26 years old, his initial presentation of hemophilia came after a tooth extraction, which landed him in the ICU requiring multiple transfusions. And kind of an interesting aspect of this is apparently, according to his hematologist on the island, is that he was one of the first cases in Canada of TRALI, the transfusion-associated lung injury, back in 1966. But again, after that, he was diagnosed with his mild hemophilia B, and things have gone from there. He had had a relatively, minor few interactions with medicine up until a few surgeries, which again were problematic. But when I saw him, he had actually been working in the back of his truck unloading some wood, and very unfortunately accidentally dropped one of the logs on his left upper thigh, and he waited for a few hours, but the swelling and pain became quite substantial enough for him to be quite concerned, and he initially presented to the ER. He identified himself as somebody with mild hemophilia, and despite that, there was still a fairly substantial delay between his presentation and him actually receiving any factor, about 6 hours in fact, and he eventually received a dose that was appropriate of about 5,000 units, and he was in hospital, and they had decided that things had stabilized and he would be safe to discharge, and so he went home. Once he returned home, he very quickly tried to start doing his activities of daily living, moving around, showering, all the above, and he found very quickly that this was quite difficult, and he was basically sedentary after that. He found that the pain was getting worse, the swelling became significantly worse, and more importantly, the hematoma that was initially in his thigh started spreading and becoming quite tense in his leg. So he returned to hospital and eventually was transferred to our hospital, where this is where I became involved. When he came to the ER, they did a CBC and noted that his hemoglobin had dropped pretty substantially to about 68 without there actually being any loss of blood externally. It had all pooled in various compartments. This was, of course, quite concerning, so they transfused him, seeing the level, and also did a CT angiogram to look for any arterial bleeding, which fortunately there wasn't any. But you can see the image here of his thighs, and you can kind of tell one is a fair bit larger than the other, and this kind of just goes to show how much blood was pooling in his leg. Fortunately, there was no compartment syndrome. But again, the hematoma on his thigh was becoming quite tense. Very shortly after he presented to our hospital, we treated him with factor IX, as well as tranexamic acid, and consulted hematology to kind of get some further instruction moving forward. The next part of this became somewhat complicated because we could see that this hematoma was quite tense and painful for him, and ideally we would extract it. And I tried contacting a number of different surgeons, vascular surgery, orthopedic surgery, plastic surgery, and was initially turned away quite eagerly because, admittedly no one's all that eager to remove a clot from someone who also has a bleeding diathesis. So it took some time, but eventually I convinced plastic surgery to see him, and this is kind of how things unfolded. So again, you kind of already had a bit of a glimpse of what's coming, but just some graphic images ahead. So this is the hematoma on his thigh prior to any intervention, initially before plastic surgery saw him. You can see there's a little bit of a laceration and some oozing, but at this time things were getting progressively larger. And you can see here that this is the extent of it, how substantial this hematoma is. It goes all the way down to his toes and all the way up to his mid-back, and was also bilateral at this point. Eventually, and just in case this is unclear, he's lying on his right-hand side, and this is his back. Eventually, plastics came to see him and did an initial debridement and extraction. Unfortunately, by this time, all the skin on top of his wound had basically died and become necrotic. You can kind of see the black tissue here that is just basically has no sensation in. This is quite a deep wound, as you'll see in the next slide. It went down pretty substantially to muscle, and the plastics department felt that they had to debride quite a lot of the tissue and take off a large amount of it. And this is what you're seeing here. They've taken off much of the necrotic tissue, and eventually what was required after this was about a 3-week hospital stay and back dressing to kind of help things heal up. Fortunately, he has recovered basically fully now, with just some minor sensation issues and some minor restrictions in mobility. But this kind of can go to show what happens when when things are recognized and treated a little bit too late. So really the lessons that were learned from this are mild hemophilia can really be anything but, and the way that people respond to injuries can be variable, but again, early recognition and in this case administration of factor is quite important to help reduce any signs of morbidity and mortality that might result. An early consultation with hematology is, I think, kind of paramount because, you know, as a medical student, I certainly was not aware of the implications of mild hemophilia B and how serious things could be, and I'm sure many of the doctors that had treated him prior to me becoming involved also had very similar understandings, and potentially is why things ended up the way they did. Additionally, large muscle hematomas can be a little bit more tricky to deal with, or sneaky as we've kind of established, and require very close assessment and follow-up. And one of the things that potentially contributed to the outcome here is his early mobilization and activities at home, which can lead to rebleeding, and you know that's very important for discharge information to these patients, that they really have to be very careful and come back to hospital as soon as they feel like things are not going in the right direction, as opposed to waiting for 3 days before they return. Mr. S has now been referred to the hemophilia and hemoglobinopathy program at St. Paul's and is being followed by Dr. Jackson now and is doing quite well. I think that's largely the case, if there are any questions.
This may not be fair because you weren't involved, but do you know why there was a 6-hour delay in getting the factor?
Part of it was, so on the island, the initial hospital that he presented to was a much smaller community hospital, and there was a transfer involved to the more larger hospital in Victoria, which, you know, patient transfer network, all that kind of stuff can take kind of a long time, unfortunately, and he ended up coming to hospital and then having to be fully reassessed because there was, I think, some poor handover. But eventually the hematopathologists were contacted and everything kind of sorted itself out. But it did take some time and definitely probably contributed to how things ended.
We have another one.
Hi, my name's Sandra. I'm actually a physiotherapist with the bleeding disorder program. Thank you so much. That was a great presentation, and we've seen this gentleman afterwards, and as you said, he's doing quite well, working a lot with physio and just getting his strength back. But what I'd like to comment on is, we talk a lot about factor, and it is so important, but it's the immobilization afterwards. So I think for this gentleman, like you commented, he really, I mean, you know, we often talk about admitting patients to hospital, and this is often a case where we do want to put somebody on bed rest. Mm-hmm. So again, the factor is important, but it's what patients do afterwards. It's the immobilization. So just to comment on that.
I actually remember rounding on him every day, and he was, while in the hospital, he was very kind of upset at the fact that he was stuck in bed. Yeah. But I mean, I think we kind of saw the result of what happened when he moved too early, so people were very cautious, and his nurses were well informed that he should not be moving.
Great, thank you.
Hi, I'm Deb. I'm a nurse with the same program. I think Mike's going on. I was just going to comment, for a muscle bleed like that, rarely one dose is enough. Like follow-up dosing, particularly for muscle bleeds, is really, really important. And I hope at the conclusion of this, we might get George's permission to show his video to all of our mild patients, in addition to the clinicians, because that's a really good point to advocate for them, to understand what you're saying with CPD. Because often mild patients will do a presentation.
Well, could I say something? Drew, I really appreciated what you had to say, but I this is on.
Give me the other mic, please.
Well, everybody can hear me. But I want to underline that what I understand is that delays such as you have, one of which you've given us an example of one, is not uncommon. And it happens all too frequently. Mm-hmm. Part of that is because we have not yet got the word out to all hospitals everywhere that there are hemophilia clinics in Canada that can be contacted and consulted. So that's one thing. But the other thing is, I know that there are big delays in even major hospitals when someone with a bleeding disorder presents themselves to them early on a Saturday morning or on a Sunday morning. They can wait for a long period of time.
Yeah, I think one of the things to note is now Mr. S has what's I think called a factor first card, and it kind of runs through the initial approach and algorithm that physicians should take, especially kind of identifies that there's this large risk, and if you don't give factor early, which is kind of the mantra of this whole early part of the talks, is the outcomes can be quite severe, and we can start to see these big bleeds and these outcomes like we're seeing.
Thank you for all of those comments. I think it's always a challenge to, we try and bring as many medical students and residents into our clinic as possible, but it's a daunting effort to try and have everyone impressed upon. And so sometimes in BC, we try a number of things to address, including advocacy and help in ensuring our patients have the Factor First card, that they know who to call, because the reality is that sometimes they can say all the right things, do everything correctly, even have their family involved, but it just doesn't work out. So often we'll have patients actually calling into the hemophilia program themselves before they get to the emergency room or en route, so that we can become involved. And we didn't get into the specifics of the factor ordering, but I think this was a patient who was not followed, like many patients in the province, by our hemophilia program. There's many more out there like him who have an event and then it's a crisis because they don't have the logistical support of the clinic. And so we're really welcoming all our patients with mild sneaky hemophilia to come into our clinic so that they can have the options of maybe having things go a little better. I think in this case we probably would have had a number of conversations with our physiotherapist, our nurse, our patient on the phone, and really tried to guide around the injury. Whenever you treat a hematoma like this in a delayed fashion, it's always going to be complex and tricky, but if we can get ahead of it a little bit, we can decrease the hospitalization, definitely decrease the amount of factor utilization that is necessary, and most importantly, help the patients with a faster rehabilitation so they're not missing work and school and other commitments in life. So it's going to be a multifaceted effort, I think, in terms of increasing awareness, but also connecting with the hemophilia program. So,
definitely.
Thank you.
Just to follow up, 2 points. One, just to your point about educating, it's really important, I guess, obviously, to educate our nurses in the emergency departments that they're the ones who make decisions, and unless things have changed dramatically since I worked in the ER, they're the ones who are going to decide, hey, intern, call a hemophilia doc to get that patient seen fast. All right, that's one thing. And the second, just to underline the comprehensive nature of hemophilia care, I think here's a perfect example. We have all our nurses and physios and our physicians. It's not just giving factor. So, and they're obviously very critical to the care of these individuals, not to mention all other disciplines, but really highlighted here. And I think we have a spectacular program here. Yes?
Yes.
All right.
Great job. Thanks.
Thank you very much. So our next speaker is Gao Arapelli, who's an associate professor of medicine and hematology at Duke University and the medical director of the pheresis unit at that institution. Gao has a great interest in immune-related causes of thrombosis, in particular heparin-induced thrombocytopenia. We had the pleasure 1 year ago of having Doug Seams here to talk about HIT, and I know that Gao and Doug are close collaborators. So Gao has come this year to finish the story and to solve the puzzle. Isn't that right?
Yes.
Right.
Thank you, Ed, and I'd like to thank the organizers of the symposium for inviting me here today. It is quite an honor and privilege to be speaking in Dr. Davie's symposium. While I did not train with Dr. Davie, I consider myself an academic descendant as I trained with Walt Kissel, the late Walt Kissel, who many of you know as a quite successful graduate of Dr. Davie's lab. So I'm going to be switching gears to thrombosis, and not really even that, because I'm focusing more on how this disorder starts in the immune response to heparin therapy. But before I get started, I always like to start off with my acknowledgement slide first, mostly because I don't want to run out of time and not cover it, and also to recognize the people who really make my work possible. And chief among the lab members and whose work I'll be presenting today is Dr. Sanjay Khandelwal, who's pictured here. He is a immunologist, very talented scientist in my lab, who made some of the seminal observations that I will be showing you today. As well, my collaborators at Duke in the Department of Immunology, Dr. Michael Frank, who is a recognized expert in complement, Dr. Kelso in B-cell biology, and my longstanding collaborators, Dr. Ponce and Dr. Seens at Penn. And of course, none of this would be possible without the support of patients who give us purpose for the work that we do, as clearly illustrated by Mr. Stevenson, as well as who provide generously their blood and tissues for the studies that I'm going to show you today. And of course, funding is essential for the work we do, and I'd like to thank the NIH and the FDA for the studies. So heparin-induced thrombocytopenia, for the audience who may not be familiar with it, is a very common disorder in hospitalized patients receiving heparin. It's an immune-mediated complication of heparin therapy, occurs in 1 to 3% of patients who get the drug. Typically, like most drug-induced reactions, occurs after a delay of 5 to 10 days when the immune response develops. And it's heralded by a drop in platelet count, a thrombocytopenia, that indicates that the immune response is underway. And really the concern about it is not the thrombocytopenia, because it's very mild to moderate, but the thrombotic complications that ensue that lead to life and limb-threatening thrombosis, as shown here. And what has been interesting about this disease, I started off studying the thrombotic complications, but as I got into work in this disease, what I realized was that the immune response to PF4 heparin occurs far more commonly in various disease settings than the actual clinical disease. And in up to 50% of patients actually exposed to heparin during cardiac surgery, they develop antibodies. And really what we have focused on in the last 10 years in my lab is really what drives this immune response. And just to give you a little bit background, the antibodies in heparin-induced thrombocytopenia are caused or directed to antigenic complexes of platelet factor 4, which is a cationic protein released from activated platelets, released in massive amounts at sites of clotting. And PF4's main biological purpose is to neutralize heparin-like molecules on the endothelium through charge neutralization. When patients with clots come in who have lots of PF4 on their endothelium, they get heparin to work as an anticoagulant. And heparin happens to be the most negatively charged carbohydrate or drug that we administer to patients. And what happens is when heparin is given, it lifts PF4 into circulation through charge interactions and form these ultra-large complexes that we have shown become immunogenic in mice. And as I said, our interest is in understanding what drives this immune response. So about three or 4 years ago, we had this very kind of an incidental observation in the lab that kind of drove a new direction for us and the work that I'll be showing you today. It started off with this observation, which was when you take PF4 heparin complexes and add it to whole blood, and we were looking for something else when we went down this path, we wanted to see what cells bind to PF4 heparin complex, and thinking that it was monocytes and that they would be processing the antigen. And as the flow cytometry is shown here, CACO is our monoclonal antibody that recognizes PF4 heparin complexes specifically, and on the x-axis are the various cell surface markers shown. And what we noticed was that really B cells bind PF4 heparin complexes remarkably and very quickly when you add the antigen to whole blood. And I mean, this antibody binding, this is just a column representation of these flow panels. This binding is wholesale. It's to practically all the B cells in whole blood, does not bind to T cells, B cells, etc. And this was just one donor shown here, but if you look at multiple donors and then you do the appropriate controls of adding buffer, PF4 alone, heparin alone, no binding. It really is PF4 heparin antigenic complexes. And so we went on to show, and this is published work, we went on to show that this is indeed happening in patients who are getting heparin therapy. And of course, patients have their own endogenous PF4. We're not adding PF4 to the blood. This is what they're generating in situ when they get heparin. And you see binding of PF4 heparin complexes to B cells in patients receiving heparin. And we studied heparinized patients in medical settings as well as cardiac surgery settings, and sure enough, we have about a 30% of patients that we could identify B cells that were transporting this antigen. And then the issue is how? How is this happening? And long and short of it is it's through complement activation. What we discovered was when you add PF4 heparin to whole blood, you're getting complement activated, and complement fragments get deposited on PF4 heparin complexes and bind to B cells, as shown here with PF4 heparin on the y-axis and anti-C3. And this is in a healthy donor, and you could demonstrate similarly that you have B cells that are coated with, that co-localize PF4 heparin and complement in patients who are getting heparin therapy. And then we showed that this binding of PF4 heparin and complement-coated antigen occurred to B cells through the complement receptor 2, CD21, on B cells. So what we showed was that if you take the whole blood, add PF4 heparin without any antibodies, you get this binding to B cells. If you add a control IgG, you get binding, but if you incubate the blood with an anti-CD21 before adding your PF4 heparin complexes, you lose binding. So the other thing we showed was that this activation occurs in plasma, and what Sanjay did to demonstrate this was he separated plasma from B lymphocytes and then did these experiments where he first incubated plasma with PF4 heparin followed by EDTA to quench further complement activation, or first incubated plasma with EDTA to inhibit complement in plasma, and then added the PF4 heparin complexes, and then added the cells and looked to see if antigen bound. And in the condition where when PF4 heparin complexes were added to plasma first, and that followed by EDTA, we preserved binding to B cells, indicating plasma contained the components to activate complement. Whereas if you did the converse, where you inhibited complement and then added the antigen and added cells, you lose binding, suggesting that if you inhibit complement in plasma, you lose antigen binding to cells. So this is kind of critical to some of the experiments that follow, showing that all of the activation occurs in plasma. So based on this work, this was our working model of HIT, and so what we show is that when PF4 is displaced by heparin, generates ultra-large complexes, this activates complement coats the antigen, and then this lands on B cells via CD21. And what the immunologists long ago in the early 90s demonstrated was if you have complement-coated antigen binding to CD21 on B cells, this is a potent indicator to the adaptive immune system to make antibodies. So binding of complement-coated antigen to CD21 enhances the immunogenicity by 1,000 to 10,000 fold. So we kind of know that we're on the path to understanding how this process initiates the immune response. So obviously a lot of unanswered questions. This is kind of the 10,000-foot view of how this process happens. And so what I'd like to talk to you about today is this part of the model that we have recently worked out in this unpublished data. So the mechanism of complement activation by PF4 heparin complexes. And so to understand these, to understand the mechanism, we kind of had to invent our own assays because none of the standard complement assays using depleted complement factors worked in our hands. And so Sanjay, went back to this simple model where you add PF4 heparin complexes to plasma and then incubate for an hour and to activate complement. And then we took this plasma containing complement-coated antigen, added it to microtiter wells coated with our monoclonal antibody, CACO, and came in with a detection antibody to detect complement that's bound to the antigen. So this kind of simplified the process and made it very easy for high throughput analysis. And this is just to show you how the data looks if you do this immunoassay with PF4 heparin. And what we also described was that this holds true for any kind of protein heparin complexes that form these ultra-large complexes. So protamine heparin is another a similar antigen that behaves similarly to PF4 heparin, causes antibodies in patients who get protamine heparin in cardiac surgery. And so we showed that complement activation also occurs with protamine heparin complexes using this assay. So one of the first things we observed, because we're constantly testing blood from people in the lab and else around us, we noticed that there was significant complement differences in complement activation among healthy donors. So shown here are about 10 healthy donors whose plasma we tested in this assay, and you can see that in this assay with complement activation detected by anti-C3c, you get nothing with when plasma is incubated with buffer or platelet factor 4, but when you add PF4 heparin, there's a striking variability in complement activation. And what was really interesting was that these differences were stable over time, and this is on the x-axis I'm showing you days where we tested these individual donors. Yours truly is shown here in red, and what you notice is that donors either had kind of a high complement activation phenotype or a low or intermediate, as shown in this graph, and the phenotypes remain stable. And so finally we had a lead where we thought, okay, this really does suggest that there's something in the plasma that is a fixed property, and surely it's one of the complement and complement regulatory proteins. So after spending about thousands of dollars on individual complement kits, we decided, okay, let's simplify this process, let's do proteomics, we can get more bang for our buck. And this table is intentionally shown here to not be read, but to show you the amount of proteins we screened. And the bottom line is that there was no correlation in complement proteins or complement regulatory proteins with the complement activation phenotype of high and low that we look for. But the money was not lost, was well spent, because we found that IgM levels differed among donors. And so this is just what we got, the data of protein intensity shown on the y-axis by proteomics, and divided among high, intermediate, and low donors. If you put it into a graph form, this is the correlation, 0.89. It's a very, very striking, significant p-value. If you did the same thing with protamine heparin complexes, which we know activate complements, same kind of linear correlation with IgM levels. And if you compare the anti-PF4 heparin and anti-protamine heparin complement activation profile, it's very linear. It's the same thing. It's all driven by IgM. And if you looked at other antibodies, and this is a larger cohort of patients, we have about 29, not patients, healthy donors, where we screened them with complement activation on the y-axis and IgM levels or IgG or IgA on the x-axis, you can see the correlation holds up with IgM, but no correlation with IgG or IgA. So clearly IgM is involved in this, and it's a correlation, but not a causation. So then we asked, is IgM necessary for complement activation in plasma? So to answer this, we had the convenience of having some donors who are very low complement activation phenotypes in our cohort, and so we took these low phenotype donors and added IgM back to their plasma. And as you can see here, these are donor 1 is green, donor 2, and there was a dose-dependent increase in complement activation when IgM was added back that was not seen with a monoclonal IgM shown in squares here, or if you add back IgG, and even if you add 5 times the amount of IgG as IgM to account for the pentameric structure of IgM, you still were not getting any complement activation. So this was clearly a unique property of IgM leading to complement activation. And then if we depleted IgM from plasma of donors who activated complement, this is shown here as a donor where we did not deplete IgM and added PF4 heparin, you could see complement activation. And if you deplete the IgM from this donor, you would see you lose all complement activation. Now, if you took plasma, added IgM like we did here, you enhance the complement activation. So if you take the depleted plasma, add back IgM and PF4 heparin, you get this complement activation profile here. So this really does show whether you add back IgM or you take away IgM really makes a difference in terms of complement activation by PF4 heparin complexes. So the next thing we wanted to know, of course, IgM is implicated in the classical pathway complement activation. We wanted to just really finally prove it by inhibiting the classical pathway. And so C1 esterase inhibitor inhibits complement activation by both the classical or lectin pathway. And if you pre-incubate plasma with C1 esterase inhibitor, prior to addition of PF4 heparin, you lose binding as shown here. And this is the control before without C1 esterase inhibitor. So this really does point to the classical pathway, but because lectin pathway also, the C1 esterase inhibitor also inhibits activation by the lectin pathway, we really wanted to be sure that it was classical. And so we did this experiment where we inhibited C1q complex with a monoclonal antibody that recognizes the globular heads of C1q. And we did this assay with incubations with dose-dependent dose-dependent levels of anti-C1q, anti-MBL, or an isotype control. And as you can see here, without any of these antibodies, you have complement activation here, and then with the addition of with anti-C1q antibody in a dose-dependent way, you lose complement activation. So this is our working model of how PF4 heparin complexes activate complement in plasma when you have just circulating PF4 at whatever level. It really doesn't do anything. You have polyspecific IgM, C1q complexes in plasma that really don't get activated. But the minute you add heparin, generate these ultra-large complexes, these then bind to IgM, probably through very low affinity but high avidity interactions, given the size of these complexes that in vitro can get to the nanomolar to ultramicron ranges, these complexes then provide a surface for circulating IgM to bind, and IgM binds, changes conformation, and activates complement by the C1QRS pathway. So in summary, what we showed was that PF4 heparin complexes activate complement by the classical pathway. I did not go into some of the specificity data to show that this is really due to polyspecific natural occurring IgM, not immune IgM, and that this activation can be inhibited by C1 esterase and C1q blocking antibodies. And what we also identified was that there was a significant donor variation in complement activation, which remains to be stable over time. And we're going to follow some of our healthy donors, hopefully for beyond months into years, and we will see truly how fixed this phenotype is. Clearly the levels of IgM determine the susceptibility for complement activation to PF4 heparin complexes, and potentially suggest that this could be a biomarker for people who may develop heparin-induced antibodies. And certainly we now have targets of the classical pathway to suggest that this could be a source for inhibiting antibody formation. And with that, I conclude. Thank you for your attention.
Question?
Sorry. Okay, so I've been involved in this for quite a long while. One of the things that we did inadvertently before we got the serotonin release assay was to inadvertently re-challenge patients with heparin. And they didn't get heparin-induced thrombocytopenia. So I'm not an immunologist, so you have to expand on that a bit for me.
Yeah, so that's another unanswered question. We don't think that there is memory so far no one's really demonstrated that there's an anamnestic or a memory response in HIT. And part of it, maybe it's a moving target. It's hard to, because this is a T-cell dependent IgG response, so it's hard to believe that there are no true memory cells that are formed. But the thing is that this is such a moving target, you have to have the right sizes complexes to generate complement and initiate, that pathway. So if you don't have enough PF4 on subsequent heparin challenge, you may not create the right kind of antigen for the B cells to get triggered. Or it could be a non-memory dependent pathway that this whole complement process initiates. These are ongoing questions we have in future studies.
Gao, that's very interesting. Thank you. So do I understand the proposed pathophysiology correctly? And that is that you make KKO-like antibodies because you bind the complexes to B cells through complement. Is that what you're...
That's exactly what we think.
Okay, so you're more likely to make an antibody, and therefore more likely to get hit if you have more IgM.
Right. So the IgM is really in the afferent arm of this whole process.
Right.
That's what it takes to kickstart the immune activation process. And that leads to, germinal center B cell responses and subsequently to IgG production.
And then once you have the hit antibody, then it does something else.
It does something else.
Yeah, and do you know if this process happens to bind these complexes to endothelial cells?
That's a good question. We have not looked at that.
Thanks very much for that. I'm wondering if CD21 variants might explain some of these, why some patients get it and others don't.
Yeah, that's a good question. We know that there is also kind of a variation in CD21 responses. We have not looked at CD21 levels in our patients, but we have focused on the plasma because right now we can show significant donor-to-donor variability just in plasma without any CD21 around. And so that's kind of where we've focused our initial attention to show the mechanism. And now we're going to go to CD21 and B cells because I think that's where the rest of the story is in terms of how that leads to antibody production and transport of antigen.
Thank you.
So, Gao, that's great. So just to follow up on Jose's question then, so once it's it's triggered by the IgM, and then you get the KKO-like antibody generated. So then do you need the IgM then to continue on with the HIT phenomenon?
We haven't looked that far yet, yeah. So we've been focused just all in, the hours to days right after heparin exposure. We have not studied what happens to IgM and IgG once you make those antibodies. But those are also questions worth pursuing.
Right, because then it would raise the question, will the C1 inhibitor, for example, as a therapy, if that was a therapy, will it work after HIT has started? Because
Yeah.
at that point,
Yeah.
the things have already taken off.
Right. And so
don't know.
where we would target it would be to try and prevent HIT. Right now, there are no alternative therapies for cardiac surgery, and about half of those patients develop antibodies. And even though, the HIT is only in a fraction of those patients, the problem is those antibodies confuse diagnosis in patients in that setting, because a lot of those patients develop thrombocytopenia for other causes, and they're treated as if they are HIT with these very potent anticoagulants, so that leads to further complications. So our thinking is that if we could even just prevent that immune response from happening, then we could remove HIT off of the table.
And there's a question way back here. You had to be so far away, Jay? I'll meet you halfway.
Excellent talk. Actually, one question regarding the immune response and PA4 heparin complex. So have you looked at the size of the complex in a way actually which, is there any correlation between the size of the complex and the immune response?
Yeah, that's a very good question. I think the size and charge are very critical to complement activation, and I think that's how IgM binds to these complexes. And so I don't have the data here, but we did a series of experiments where we looked at the types of particles that are formed between PF4 and heparin. And the nice thing about that is that it's all based on the stoichiometric amounts of PF4 heparin. So if you have just nanogram per ml quantities of PF4, you make small complexes. The more you add of PF4 and heparin, the larger those complexes get. And it's just through colloidal interactions, they just aggregate and make big complexes. And so if you add small complexes with high IgM, you activate. Or if you have, large complexes with low IgM, you get activation. So there is a relationship between the size of the complexes and the amount of IgM you have in terms of whether you're going to activate complement or not. Is that what you're asking?
Any other questions? If not, Gav, thank you very much.
Thank you.